Hello friends, we will be discussing the project that we developed for IT 468 Natural Computing course offered in DISCT. So in this project, we basically had developed add-on features for this DNA Pen software, which is developed by Gupta Labs. So DNA Pen software is basically a user-friendly interactive environment for the users to envisage their DNA structures and get the actual DNA sequences required to make the structures physically. Okay, so the add-on features or the source code is available in the GitHub repository, as you can see. Okay, so the source code is available. Also, we are supplying basic libraries which you would be requiring so as to run the software. So these are the libraries which you need to include along with the software. Also, we have supplied some test images for which you can run the software. Okay, so once you open the software in Eclipse, you will have to import the whole software over here. So as you can see, we have imported all the Java files. Along with this, you will also have to include all the libraries, as you can see here. Okay, cool. So now let's start with the demonstration. So once you run this software, you will first have to create a project, which you can do by creating a new project over here. As you can see, initially most of the options have been disabled, so you cannot use them unless you create a new project, right? So the tools have been disabled, edit has been disabled. So you go to file, you create a new project. Let's name it something ABCD. Create. Okay, so first you have to choose a location where you want to create. Okay, create the file. Okay, so a project has been successfully created. Next, now what you can see is that some of the tabs have been enabled now, and you can go use the various options available. So there are two options available now. One is freehand molecular canvas and the other is digitized molecular canvas. Okay, so first let's talk about freehand molecular canvas. So in this, what you can do is you can basically draw an image over here, and once you save it, it will give you a DNA sequence required for drawing that image, right? So in earlier software, you had to manually draw the image, right? So you have to create something called as like an ohm. So it will be a very tedious job, like to use this and like do it all manually. So, and also erasing it is a bit of problems, right? So it takes a lot of time. So what you can do, the new feature that we have added now is you can go to the tools menu, and now you can see that the import free image has been enabled. The grid image is still disabled because you're working in the freehand molecular canvas. So you go to import free image, and let's try to import some image, right? So let's try to import a new ohm first. Okay, so as you can see that it actually drew the ohm which we had already drawn in some file. So let me show you how the file actually looks like. Okay, so this is our ohm which we had already drawn, and as you can see, it has now drawn it on itself on the canvas, right? So it saves the job of drawing all things manually. Next, you can go to save detailed DNA data. Okay, so it as the file has saved successfully, which means that all the DNA sequences have been saved. And you can go to like view draw image, where you see what kind of the DNA structure will be formed and the sequence required to create this image. Okay, cool. Additional feature to this is something like this. So let's try to import some other image now, and let's try to import a ohm which is disconnected, right? So as you can see that this image is not connected, so this line is different from this ohm structure, this is different. So it will give you a new warning saying that the image is not connected and please draw a connected image before saving it, right? So this is important because otherwise if you create the DNA, so the things will be just floating in the test tubes. Okay, so it needs to be connected, so it gives a warning regarding this. Okay, so you can also have complicated images like I'm going to import free image, just right? So it actually makes a whole map of India now. right? So you can work with many images and you can save them. You can get the DNA sequences required to create those images. Okay, so let's now start with some new. So other thing that you can work with is the digitized molecular canvas. This is a brick-like structure, like in which if you press the brick, which means that it is enabled, you can disable it by again clicking it. So again, if you want to like draw something like ohm, so you'll have to like click all the bricks so that you make something similar to an ohm. So which is very tedious, right? So what you do, you just go to tools, and now you can see that the import free image has been disabled, and the import grid image has been enabled, right? So you go to import grid image. Let's try to import some image, right? So there's 2 new ohm, and now you see that okay, so your ohm has already been drawn, right? So it's still not very accurate, but you can just like select and deselect some bricks so as to refine it even more, right? So you can have like something like this, right? And maybe just remove this, right? Maybe have this, maybe remove this. Right, so it more resembles like ohm now. So you'll have to do some fine adjustment, but the initial work of like clicking all the bricks has been removed, so it is all automated now. Again, you go to save detailed DNA data. The file has been saved, and you can see how it will look in the view draw image, right? So this is how it will look in the DNA form, and these are sequences required to get this image. So this was all the new features that we added to the DNA Pen software. The source code is available on GitHub, so you can download it from there, right, by going on to the repository. We would be sharing the repository details in the description below. Do like, share, and subscribe. Thank you.